To Bio 216. I'm Udra Kaleton, your facilitator. In today's video, we are going to continue with monosaccharides. We'll look at derived monosaccharides and we'll also consider some reactions of monosaccharides. Have a nice time as you watch the video. So this is Unit 3, Bio 216. And well, like I said, we'll look at some monosaccharides and then some properties. At the end of this lesson, you should be able to at least list some monosaccharides other than glucose, explain three types of derived monosaccharides, and then explain some of the chemical properties of monosaccharides. So, apart from glucose, other monosaccharides also exist. And these monosaccharides are named according to the number of carbon atoms we have in them. So when you hear triosis, you're talking about monosaccharides that have 3 carbon atoms. And examples include glyceraldehyde and dihydroxyacetone. Just like glucose, just like 6 carbon monosaccharides, also known as hexoses, can either be aldehydes or ketoses. The same also occurs in 3 carbon monosaccharides, tetroses, pentoses, hexoses, and heptoses. They can either be aldehydes or ketones. So for tetroses now, erythrose is an aldehyde, whereas erythrulose is a ketone. It's a ketose, sorry. Ribose is an aldehyde, whereas ribulose is a ketose. Galactose, mannose, and allose are aldehydes, whereas fructose and these are ketoses. L-glycerol D-mannose heptose is an aldehyde, whereas pseudoheptulose is a ketose. Let's look at derived monosaccharides. There are different types of derived monosaccharides. We can have sugar acids, oxy sugars, amino sugars, alcohol sugars. Like I said, derived monosaccharides. That means these monosaccharides may be due to a change or an addition of one functional group or another. They become whether sugar acids, deoxy sugars, or amino acids. Sorry, amino sugars. So when the carbonyl group or the hydroxy group is oxidized to a carboxylic group, you have a sugar acid. And this can occur in various places. If it occurs in carbon 1, for example in glucose, it becomes gluconic acid. If that oxidation occurs in carbon 6, on carbon 6, it is called a uronic acid or glucuronic acid for glucose. At times, a very strong oxidizing agent might oxidize a monosaccharide, for example glucose, on both carbon 1 and carbon 6. If this occurs, we have an aldaric acid. For glucose, it is called glucuric acid. So that's it. At times, we may also have deoxy sugar. As you can see, deoxy. What does that mean? This is the normal ribose. You see this now? And oxygen has been removed, so it becomes a deoxy sugar. And now you have this one, 2-deoxyribose, meaning that the oxygen on the OH group of carbon 2 has been removed from this ribose. I'm sure you must have heard about DNA and RNA. This is one of the differences between a DNA and an RNA. When when we have removed the oxygen from the ribose that should have been OH like this, you say you have a deoxy. So this will now the part of the sugar part of the DNA is now lacking one oxygen, so it is deoxyribose, whereas this, as is found in RNA, is called ribose, deoxyribose and ribose. Another form of derived monosaccharide we have are called amino sugars. Simple, just as the name appear just as the name tells us. The only difference is that there is an amino group attached to one of the carbon atoms there. In this case, in this structure we have, is attached to carbon 2. You can see it is still, every other thing is the same apart from that. So, and because this amino group has been attached to this sugar, it becomes an amino sugar. So it's a class activity. You remember what we talked about in glucuronic acid. Now I want you to draw the structure of a glucuronic acid. From our explanation, how many carboxylic acid groups do you think this will have? Good. So draw it. Now let's go to the next part of our class. Now we're looking at the properties of monosaccharides. Properties of monosaccharides. What are the properties of monosaccharides? Monosaccharides undergo mutual rotation. They have reducing property. They can form glycoside. They can form esters. They can be dehydrated, and then they can also undergo rearrangement in alkaline solution. So what do we mean by mutual rotation? When you talk about mutual rotation, we must consider our anomass. You remember when in our last video, in our last lesson, we were talking about anomass of glucose. When a glucose becomes cyclic, we said that when the OH on carbon 1 is down, it is an alpha glucose. When it is up, facing up, it is a beta glucose. What is mutual rotation? Mutual rotation is just a phenomenon whereby alpha and beta glucose can be converted in a glucose solution. So if you have a solution of glucose, what we expect to have 36% of the molecules as alpha glucose, less than 0.1% as straight chain and 64% as beta glucose. So that is it about mutual rotation, just interconvertibility of the alpha and beta anomers. Then the reducing property. Monosaccharides readily reduce oxidizing agents. Monosaccharides readily reduce oxidizing agents. And this is what the principle that applied when you are here, when we want to do test for glucose. For those of you who must have had at one time or the other a relation who is diabetic, say, you have to go and test the glucose in the, you have to test the glucose in the blood or glucose in the urine. Is the reducing property of glucose that is applied. What happens? Under normal, when glucose reacts with an oxidizing agent like copper 2 ion, the glucose tries to reduce the copper 2 ion. That's why we say it has a reducing property. It reduces the copper 2 ion to copper 1 ion. And in a solution, if you have a solution, maybe a solution of glucose, and you boil it and introduce this agent, for example, Benedict solution containing copper 2 ion, you will have a brick red color. This Benedict solution at times is also applied in quantitative tests for glucose. In that way, when I say quantitative, it's from the word quantity. It's trying to tell you how much glucose is in that place. So assuming we put it in, we test somebody's urine and it gives us a brick red precipitate, you say, that the glucose level is so high. Whereas if it gives us something like this light, this greenish yellow, okay, at times we say dirty green precipitate, you say, okay, not too much. Or this orange, no, but it's not, when it's orange, it's not too much. But when it is brick red, the person's glucose level is high. Also, we can use this Benedict, we can also use Benedict solution and for qualitative test. In other words, when you talk about qualitative test, just telling you, okay, it is there, but it doesn't tell you how much it is. So it is the reducing property of glucose that is involved, is applied when we're doing this test. Another property of carbohydrates, monosaccharides, is that they form glycosides. What happens when the glycosides are formed? When hydroxyl groups react with the anomeric carbon atom. When hydroxyl groups react with the anomeric carbon atoms, and most times we can get this hydroxyl group from alcohols. So when they react, they form a compound known as glycosides, a group of compounds known as glycosides. There are so many types of glycosides in nature, and they have different functions. A glycoside formed from glucose will be called glucoside. Then also, monosaccharides also form ester. I hope you remember from your chemistry when we talk about the process of esterification. What do we mean by that? When a hydroxyl group reacts with an acid to form a compound, the compound formed is known as ester, and the process is known as esterification. We don't need to talk about, we don't need to say where will monosaccharide get their own hydroxyl group. Remember when we drew the structure of glucose, we saw a lot of hydroxyl groups present. So that's why glucose or monosaccharides are able to get involved in many types of reaction. They have those hydroxyl groups, and those hydroxyl groups are very reactive. They also have the carbonyl group. So that's why you can see that glucose can be involved in many types of reactions. So also another type of reaction, monosaccharides, and really not just monosaccharides, really most carbohydrates undergo this reaction, the dehydration reaction. What happens when you add a strong acid like HCl to something like pentose or hexose? It dehydrates to form fluoride or hydroxy methylfluoride. If that compound is not maybe a hexose or pentose, it first undergoes hydrolysis and then it still forms that. So this smallish test is a sure test for carbohydrates. You see from the diagram we have here, when the carbohydrate is positive, you will see a bluish or purplish ring in the test tube. In the Molish test, apart from this strong acid, we also add something we call alpha naphthol, but I don't want us to go through that now. Just understand that the hydration reaction of monosaccharides or carbohydrates in general is the principle employed in the Molish test for carbohydrates. So also, apart from the fact that they are reducing agents, monosaccharides too can be reduced. They also undergo reduction. Monosaccharides also undergo reduction. When a mild reducing agent attacks monosaccharides, they form alcohols, or we call sugar alcohols. And this sugar alcohol is employed in a lot of industrial processes. I'm sure most of you must have chewed gum. I don't want to like Orbit, the chewing gum called Orbit. That chewing gum, they will tell you low sugar because the sugar in it is the alcohol sugar. Alcohol sugar does not have the effect. For example, dentists will tell you that this one does not um, um, cause tooth decay because the sugar there is now an alcohol sugar. And apart from the fact that it doesn't cause tooth decay, it also does not have as much calorific value as normal sugar. So that's for alcohol sugar. So let's take another class activity. I want you to list 3 chemical properties of monosaccharides. I'm using chemical properties now because in our first lesson we looked at the physical properties of monosaccharides. For example, when we say monosaccharides are white, they are solid, they are sweet. Now let's look at the chemical properties. So I want you, and we have mentioned some today, I want you to list 3 of those chemical properties we have mentioned. Good. In today's class, we have been able to show that apart from glucose, monosaccharides can also exist in other forms. having more 3 to 7 carbon atoms. We have also seen some derived monosaccharides like sugar acids, amino sugars, and deoxy sugars. And I also told you why they are called, what they are called. And then finally, we looked at the properties of monosaccharides, their ability to form reducing sugar, their reducing property, their glycoside formation, ester formation, among others. Thank you.